Okay, well, good afternoon, everyone. Thank you so much for joining us. We have a lot of really important information to share with you today, so we're really glad you're here. For those of you who don't know me, my name is Monica Coonrads. I'm the founder and CEO of the Rett Syndrome Research Trust. My inspiration and my motivation comes from my 27-year-old daughter, Chelsea, who was diagnosed with Rett syndrome at the age of 2. For the past 25 years, hard to believe that it's been that long, I've been raising research funds and working with the scientific and clinical community to advance therapeutics for our children. When I started, we didn't know that MECP2 mutations caused Rett. Very few scientists were working on the disorder or even knew about it. There was no industry awareness at all, and genetic medicines for Rett was just wishful thinking. We've come a very long way since then, and I'm excited to tell you about our most recent progress. Okay, let me share my screen. So I'm very, very fortunate to have amazing colleagues who are just as passionate as I am about curing Rett syndrome. Bob Deans joined RSRT a year ago in a newly created position, Chief Technology Officer, Head of Research. Bob has decades of industry experience moving genetic medicine programs from the research stage to the clinic. Adding Bob to our team has been a game-changing move for RSRT, and we are thrilled to have him. He's joining us today from Riverside, California. Jana Van Heen is our Chief Scientific Officer, Head of Clinical Development. She joined us 7 years ago. Jana has over 17 years of experience in clinical trial operations, databases, and program management. And she puts all of that great experience to work, heading up all of our clinical research initiatives. Jana lives in the Boston area, right where all the biopharma action is. Tim Freeman is our Chief Development Officer, which means he's in charge of our fundraising effort. Fundraising and research progress go hand in hand. You can't have one without the other. Tim has been with RSRT for 11 years. And like me, he's motivated by his daughter, Eleanor. And he's joining us today from Princeton, New Jersey. Completing the RSRT team are a few people that aren't on the webinar today. Randy Carpenter, our Chief Medical Officer. He's an expert in translational medicine. That's the process of turning laboratory discoveries into interventions that actually help people. He's been with us for 9 years, and we're extremely lucky to have someone of his caliber working on our children's behalf. Jenny Reynolds, our research manager, supports the research team with emphasis on the clinical research efforts. She's been with us for 3 years. And Ricky Davis, our administrative officer, keeps the organization running smoothly. She's been with us for 8 years. Now, I'd like to just take a moment and emphasize the strength of our 5-person research team. Collectively, Bob, Jana, Randy have decades of experience. They've worked for big and small biopharma companies. Bob and Randy have launched several startups. And apart from an industry background, this team also has business development experience, which has become so critical given the stage of our science. Now, earlier this summer, we unveiled Roadmap to Cures 2.0, our strategic plan for how we advance genetic medicines for Rett. I think it's widely known that RSRT's entire focus is on genetic medicines. And the reason for this is our firm belief that dramatically improving Rett symptoms will require targeting the root cause of the disease. Now, here's what we're going to cover today. We're going to introduce Roadmap to Cures. We're going to explain what it is and why we need it in the first place. We'll take you through all of the various genetic medicine approaches that we're advancing. We'll explain which genetic medicine approach is relevant for which mutation. We're going to provide a timeline for when we expect to be in clinical trials. We'll also discuss the clinical research initiatives that Jana is spearheading We'll talk about the cost to advance these programs to the clinic and how you can help. And please make sure you stick around to the end, because we're going to have a frank discussion about the C word, cure, what that means, how we're thinking about it, and what it means for your loved one. We'll leave plenty of time for your questions. And by the way, please feel free to add your questions anytime into the Q&A, and we'll answer them at the end of the webinar. Now, genetic medicines are made up of two components. The cargo, which is what you're actually trying to deliver into the brain. So for gene therapy, for example, that cargo is going to be the MECP2 gene. But for other approaches, the cargo will be something different. The other equally important component is the delivery technology, which means how that cargo actually gets into cells. You can have the best cargo in the world, but if you can't get it into a sufficient number of brain cells, it's not going to do much. And it's the combination of cargo and delivery technology that makes up a genetic medicine. So the goals of Roadmap are very clear. Early next year, we will select 3 genetic medicine programs to advance to clinical trials. In order to begin a trial, we need to submit what's called an investigational new drug application, IND, to the FDA. As part of that application, we need results from a very structured set of experiments, and those are known as IND-enabling studies. These studies address safety, estimation of a safe starting dose, dose ranging studies, and more. We'll engage the FDA early and often, and the cost of Roadmap is $40 million over 4 years, so $10 million a year. By the way, there's nothing magical about selecting 3 programs. We decided on 3 because we think that's what we can manage and afford. But with additional financial resources, there will be a 4th and a 5th and more. The more shots on goal, the better for our children. Now, some of you may be asking yourself, why do we need Roadmap at all? Gene therapy trials are underway. They were decades in the making. Doesn't that mean a cure is around the corner? That's a legitimate question. So we're excited about the current gene therapy trials. Both of these programs were started and were incubated at RSRT, and we are thrilled to see them move forward. We expect that the trial participants will improve, but we also expect that there will be room for additional symptom improvement. The first genetic medicine out of the gate is just that. It's unlikely to be the best or the last. It's the first. Drug development, including genetic medicines, is an interactive process. Think back to the first iPhone, an amazing breakthrough in technology. But fast forward 17 years and we're on iPhone 15. So there will be genetic medicines beyond the current gene therapies being tested. That's a given. And that's very good news for our children. In fact, we can look to the SMA field, spinal muscular atrophy, as an example of what's to come. There are multiple genetic medicines for SMA, including a gene therapy that was approved in 2019. And yet there's a very active genetic medicine development effort aimed at improving what's currently available and offering better options. Lastly, gene therapy is one kind of genetic medicine. Today, you're going to hear about other approaches as well. We think it's important that we advance all of them in parallel. Bob, would you like to add anything?
Yeah, thanks, Monica. First, I'm real glad to be getting to know more of you today, and I'm always available to connect as a follow-up after the webinar. So our new roadmap reflects our move from funding basic science and moving to clinical translation. This community should feel very good about the quantity and the quality of basic science that has gotten us to this point. It's really exceptional. We have confidence in multiple approaches that we'll tell you about shortly, and that we plan to take to the clinic with an emphasis on improved delivery. The roadmap to the clinic is going to get done in concert with biopharma partnerships, an important concept here that we're developing as we speak. So thanks, and back over to you, Monica.
Thanks, Bob. So we're going to play for you a short clip from a very well-known scientist, Fyodor Urnov. He works for Jennifer Doudna, who won the Nobel Prize in 2020 for discovering CRISPR. a genome editing tool that has completely revolutionized research. Now, Fyodor and his colleagues actually coined the term genome editing. He attended the Rett Syndrome Genetic Medicine Summit that we hosted last September in Boston, and he said the following.
The one thing we've learned about how to build a cure for genetic disease is we have to put as many different technologies for those diseases into the clinic and learn as much as we can from human clinical trials. I'm not saying we have to try everything, but we have to do trials for every approach that has meaningful clinical promise.
So we wholeheartedly agree with Fyodor, and our plan is really to advance as many programs in parallel as our resources allow. Now, here's a concept that you may not be aware of, and it's going to make it very clear why roadmap is essential. Biopharma companies are unlikely to start genetic medicine programs from scratch. It's too risky for them. Rett is not low-hanging fruit for companies. Rett is a brain disorder, which makes delivering genetic medicines more challenging. Delivering to the liver, the blood, the eye is much easier. And those are the low-hanging fruits that companies are going to start with. So it's really up to RSRT to fill this pipeline with genetic medicine programs and to identify biopharma companies to transition the programs to once they've been de-risked. Feed the pipeline. You're going to be hearing that phrase a lot. And we're really the only ones that are doing that. Without RSRT feeding this pipeline, the pipeline dries up. And that means less options for our kids. Cures for Rett are dependent on advancing a rich pipeline of genetic medicine programs. Now, this slide visualizes the key role that we play in transitioning discoveries from academic labs to companies. Valley of death is a term that's often used in drug development. It's where great ideas go to die. And we're here to make sure that that doesn't happen for Rett. Both the Neurogene and the Taysha gene therapy programs were started and cultivated at RSRT. We selected the scientists, provided funding, launched the gene therapy consortium, and the data that was generated in this consortium convinced Taysha and Neurogene to license the programs and advance them with their own funding. Now, you can find information on all of these companies on our website, but the bottom line is that it's unlikely any of them would have programs for Rett without RSRT. Now, the companies listed here have publicly announced their Rett programs, but it makes me very happy to tell you that there are at least another handful of companies who are working on Rett genetic medicine programs who have not yet announced them. Now, before discussing the actual programs, I want to remind everyone why we are so optimistic that targeting the root cause of Rett is the way to go. First, Rett is caused by mutations in a single gene. Now, that's not the case for many other disorders. Think about Alzheimer's, Parkinson's, autism, schizophrenia, MS, and so on and so on. Having a single gene gives us a very clear target for intervention. By the way, I want to make sure that I tell you that when I say mutation, I mean any alteration to the MECP2 gene. That includes insertions and deletions, any kind of alteration. Secondly, brain cells in individuals with Rett don't die. For neurodegenerative disorders, you have to intervene early because once cells die, it's very difficult to regain function. But in Rett, all the cells are there, they're all in the right place, and they are not dying. That is very good news. And finally, courtesy of Adrian Bird, we know that symptoms are remarkably reversible in mice models, regardless of age. So combined, these three facts convince us that Rett syndrome screams out for a genetic solution. So what exactly are genetic medicines? I'm going to let Bob answer that question.
Yeah, I'm happy to. So genetic medicines are DNA or RNA that are injected into the body as therapeutics to supplement or correct the genetic code, the MECP2 genetic code, and make functional protein. So these medicines need to be delivered to individual cells in the brain, a daunting task. So effectiveness is highly dependent upon delivery technologies, cargo and delivery, and routes of administration. You're going to hear a lot more about this later in the presentation. So as I mentioned before, the field has moved out of the discovery stage. We know how to fix a defective MECP2 gene. This isn't pie in the sky. This is happening. The clinical advances you're hearing about are going to accumulate, and we're going to find the solutions that we're after. We need to ensure that our academic and industry partners keep frontline focus on Rett, and Monica set up an organization that's just been doing a stellar job at that. I'm real happy to be part of this group. Back over to you, Monica.
Thank you. Okay, so I mentioned earlier that genetic medicines are made up of two components, cargos and delivery technologies. So now we're going to discuss the cargos. Keep in mind that the end goal is the same for each of these cargos. It's to boost levels of the MECP2 protein, but each cargo does it in a different way. So we're going to start with gene therapy. That's our first cargo. So with gene therapy, we're not correcting the mutation. The mutation is still there. The mutated protein is still being made, but we know that mutated protein isn't doing anything bad. It just doesn't work properly. So with gene therapy, we are adding healthy copies of MECP2 to compensate for the mutated gene. Now, as you all know, there are two industry gene therapy trials, one sponsored by Neurogene and the other one by Taysha Gene Therapies. And you can see here from the table the ages that are being recruited, 4 to 10 for Neurogene, 5 to 8 for Taysha's pediatric trial, 12 and over for their adolescent adult trial. Now, if the results are encouraging, I would expect the age ranges to expand. Of course, that will be up to the companies to decide. Now, Neurogene's trial is recruiting in the US and Australia and expected to recruit soon in the UK. Taysha's adult trial is in Canada and the US. The pediatric trial is recruiting in the US and soon will be recruiting in the UK and Canada as well. Now, both programs are using the same delivery technology, the AAV9 vector. And we're going to talk more about that a bit later when we get to the delivery technologies. In terms of cargo, Neurogene is delivering a full-length MECP2 gene, while Taysha is delivering a shorter mini version of the gene. In terms of route of administration, how the gene therapy actually gets injected into the body, Taysha is using intrathecal, or IT, which is an injection into the base of the spine, similar to an epidural. The patient is injected and then placed head down so gravity can assist in getting the gene therapy into the brain. Intrathecal injection is a routine procedure which can be done in a doctor's office. Neurogene is using intracerebroventricular, or ICV, which is an injection, single injection, into the ventricle of the brain. Now, ventricles are cavities inside the brain that make and store cerebrospinal fluid. This fluid surrounds the brain and the spinal cord, protecting it. It also delivers nutrients to the brain and removes waste. ICV injection is done in an operating room with a neurosurgeon. And I know that it sounds a bit scary, but it's actually quite a routine procedure. Now, we know that too much MECP2 is detrimental. So both programs have a way to guard against that. Neurogene's technology is called EXACT, and Taysha's is MI-rare. Both programs are testing a high and low dose, or I should say a low and high dose of their gene therapy products. Neurogene has stated that they will be sharing efficacy data in the fourth quarter of this year, and Taysha stated in their press release last week that they'll be sharing data in the first half of 2025. Now, it's important to note that gene therapy should be able to address any kind of mutation, and it should be a one and done procedure. RSRT funded our first gene therapy project in 2010, and we've invested $11 million in gene therapy projects since then. Now, there's many different types of cells in the body. A heart cell is different than a liver cell, and both are very different from brain cells. The brain is the most complex organ in our body and has over 3,000 cell types. Now, the amount of MECP2 protein that is made in a cell is dictated by the cell type. In every cell, there are stretches of DNA that don't code for protein. Instead, they regulate protein production of neighboring genes. These stretches of DNA are called regulatory elements. So the MECP2 gene has regulatory elements that dictate how much MECP2 protein is made. Now, for a disease like Rett syndrome, where dosage really matters, correcting the native gene or the native RNA may have advantages, because when you correct the native DNA or native RNA, the regulatory elements are already present. So the right amount of protein will naturally be made in every cell type. So now we're going to discuss the actual approaches for correcting mutations. So base editing can correct mutations that are caused by a mutation or a typo in a single nucleotide base. Base editing does not naturally exist in nature. It was engineered by David Liu, a scientist at Harvard. It was first reported in 2016, and the technology is already being used in clinical trials, which is really incredibly fast. RSRT has awarded over $8 million to base editing projects. Now, base editing is not a generic fix like gene therapy. It's mutation by mutation. So a base editing therapeutic would have to be developed for every mutation. The next approach is called prime editing. And I like to describe it like base editing on steroids. And that's because it can fix multiple mutations that are physically close to each other on a gene with a single therapeutic. So that means more mutations can be addressed with a smaller number of therapeutics. And as the technology gets better, it's going to be able to span larger and larger sections of DNA. Like base editing, prime editing does not naturally exist in nature. It was engineered also by David Liu. He first reported prime editing in 2019, and trials using this technology are about to start. Again, incredibly fast and very exciting. Now, to date, we have funded just over $1 million in prime editing. You can expect that amount to increase. RNA editing does exist in nature, and it's catalyzed by an enzyme called ADAR, which can correct a certain type of point mutation, a mutation in a single base that's in particular a G to A mutation. This approach will also require a specific RNA editing therapeutic for every G to A mutation. This modality does not require delivery with a viral vector. We have funded over $7 million in RNA editing projects, which has really put RNA editing on the map for Rett syndrome. Now, these are the companies pursuing RNA editing programs for Rett. Vico is a company that was started by Gail Mendel, who we funded for over 10 years, and who together with her postdoc, John Cinnamon, pioneered RNA editing for Rett. Shape was able to launch a Rett program because they had access to our biorepository of patient cell lines, which gave them an enormous running start. We recently provided over $1 million of funding to ProQR, a Dutch company who is working closely with one of our funded researchers, Pete Beal. And I'm going to turn things over to Bob for a bit to elaborate.
Yeah, thanks. So a theme that we're emphasizing is that the ability to deliver genetic cargo into cells is critical. It's as important as the modality you're using to correct the gene. So a very attractive feature of the small RNA approach in our work with ProQR is that it doesn't require viral delivery. The small RNAs are chemically synthesized, suspended in solution, and delivered into the body with a simple injection, half-life of 6 months to a year. So not relying on viral delivery means that this approach, the small RNA approach, is amenable to redosing when required and opens up the possibility for patients to receive, in addition to the RNA editing approach, a virally delivered genetic medicine. A patient could receive a gene therapy and an RNA editing approach. This is what has been developed in treatment of spinal muscle atrophy, and we're real enthusiastic about the combination potential for some of the approaches we're taking. And back to you, Monica.
Thank you. So last year, we funded a record $10.2 million worth of research, and we launched the MECP2 Editing Consortium, which is a collaboration between these 6 labs: Adrian Bird from the University of Edinburgh, Guoping Feng for MIT, Pete Beal from UC Davis, and then 3 labs at UMass Medical School in Worcester: Eric Sontheimer, Scott Wolf, and Jonathan Watts. The labs are working on the 3 modalities that you see here. Guoping and Adrian are working on base editing, Pete's doing RNA editing, and the UMass labs are pursuing both base and prime. Bob, you had a chance to meet all of these researchers recently at an in-person meeting. I think you wanted to elaborate a little bit about.
One of the really attractive features at RSRT has been the caliber of the associated sciences. It's really impressive. I'm finally in a club I want to be in. It's really great. So I wanted to mention a couple of things. Scott Wolf is an advisor to the FDA on in vivo genome editing approaches. So we've got somebody in our camp that is really well versed on the safety and toxicity issues around the different modalities. Eric Sontheimer, also at UMass, is a co-founder of Intellia, one of the first and premier gene editing companies, which is active in vivo gene editing approaches in the liver. Guoping Feng, we're really proud to announce, is a member of 2 national academies. He's risen to be a member of the National Academy of Medicine, as well as a member of the National Academy of Sciences. And I always like to say the Sir and Sir Adrian Bird really says it all. So stellar group.
Now, all of you probably know that the MECP2 gene is on the X chromosome, and that females have 2 X chromosomes in every cell. Males only have 1. So very early in development, females shut down one of their X's in every cell. So girls with Rett have 2 MECP2 genes in each cell, but only one of them is active. In some cells, the X chromosome with the healthy MECP2 is active, and the mutated is inactive, and then it's the other way around in the other cells, opposite that. So with this approach, we are trying to wake up the silent MECP2 that's on the inactive X. This means cells would have both MECP2 genes on, the healthy one and the mutated one. And keep in mind that the mutated gene doesn't do anything harmful. It just doesn't work. We funded over $9 million on reactivation projects. Some of those funds have gone to a collaboration between the 3 labs you see here: Tony Bedilov, Kyle Fink, and Sean Liu. Now, this is a really ambitious, challenging program, in part because the editing machinery that we need to use was large, couldn't fit into a single vector. But with a lot of work, they've been able to shrink down the size of that machinery so that now it can fit into a single vector. Now the program has become much more clinically feasible. The final cargo is RNA trans-splicing. You can think of MECP2 as a book with 4 chapters. 97% of all of the mutations are in chapters 3 and 4, and the remaining 3% are in chapter 1. Trans-splicing is something that naturally happens in a cell, and I'm going to give you a brief description. So DNA, as you know, holds the genetic instructions to make protein. DNA lives in the nucleus of the cell, and it never leaves. But proteins are made outside the nucleus. So the DNA makes a copy of itself, and that copy is the RNA. And the RNA is made up of introns and exons. The introns are then removed, and the exons are spliced together. As the exons are spliced together, we want to drive the process so that the mutated exons get removed and are replaced with healthy exons. One therapeutic would address 97% of all of these mutations, and a second therapeutic would address the remaining 3%. Now, until recently, RNA trans-splicing was a bit of an obscure technology with very few people working on it. That has dramatically changed over the last year or 2. Today, there's a number of biotech companies pursuing this approach, with more on the way. We're talking to all of them, as this is an approach we're really interested in. We've invested $1.1 million to date, expect that amount to go up. So these are all the cargoes. Keep in mind that the end result for each one is the same, boosting levels of MECP2, but each cargo goes about it in a different way. Now we're going to be discussing delivery. So there are two broad categories of delivery, viral and non-viral. Viral delivery takes advantage of a virus's natural ability of getting into cells and infecting them. Scientists realized early on that they could take advantage of this natural ability by removing the parts of the virus that make people sick and using the virus as a Trojan horse to deliver cargoes of their choice. Now, the current gold standard for viral delivery to the brain is AAV9. And you heard previously that that's what Taysha and Neurogene are using. It can get into the brain quite effectively, but it's not perfect. For example, in some parts of the brain, it might be able to get into 10, 15, 20% of the cells, while in other parts of the brain, the percentage might be as low as 3 to 4%. And deep brain structures are not accessible at all to AAV9. So there's a major effort ongoing trying to find viral vectors that work better than AAV9. And there's starting to be some success in this area. My colleagues and I are working to gain access to these better vectors. Now, viral delivery does have some big challenges. It can only be administered once to patients. If it's given more than once, the patient may mount an immune response, which could be very severe and even deadly. So that means families will have to be very, very thoughtful about choosing a genetic medicine that is delivered via AAV9 or any kind of viral vector, because if something better comes along that's also delivered virally, that patient wouldn't be able to get it. So there are opportunity costs that need to be considered. Now, there is an effort to overcome this issue, and it's possible that in time, scientists will figure out how to redose a viral vector. Now, the other challenge is that viral delivery has a very complex and expensive manufacturing process. Gene therapies are expensive, $2, $3, $4 million for a one-time dose. But in large part, that's due to the very high cost of manufacturing them. Now, because of these challenges, there's a huge effort underway to develop non-viral delivery technologies that access the brain. As the name implies, these are delivery technologies that don't use viruses, and there's lots of different ones. For example, lipid nanoparticles, LNP. That's how the COVID vaccine is delivered. The messenger RNA is enveloped in a little fat bubble. LNPs naturally want to go to the liver. So genetic medicines aimed at liver diseases are moving away from viral delivery and using LNPs. And there's a big effort to engineer LNPs so they can get into the brain. That would be a huge breakthrough. But no one's cracked it quite yet, and many are trying. Another type of non-viral delivery is RNP, ribonucleoprotein complex. We just awarded funding just a couple of weeks ago to a scientist at Yale who's developing RNPs, and he'll now be working with 2 of our researchers on the cargo side. Check out our website later this week for more information on these projects. Now, the advantage of non-viral delivery is that there's no immune issues, so they can be redosed. A patient could theoretically be able to access multiple genetic medicines, and they're cheap and easy to make. So our plan in terms of delivery is to move ahead with viral vectors for our current projects, to access novel vectors whenever possible, to stay on top of the non-viral delivery landscape, invest wisely in these technologies, and when possible, make the switch from viral to non-viral. Bob, anything to add?
Yeah, I really place a lot of emphasis on reducing cost of goods and ensuring that we can get broad accessibility to patients worldwide. And Monica mentioned the cost of a dose of an AAV vector in the range of $1.5 million. You can imagine what that represents to a smaller biotechnology company in terms of development cost and being able to practice at scale. It can be prohibitive in terms of making a commitment to treating a CNS-related disease. So I'm really excited to see us moving to synthetic lipids and then even to the core basics of the small RNPs. It's estimated to bring our costs down from $1.5 million per dose to something in the range of tens of thousands of dollars, with repeat dosing being feasible. So it's really a sea change opportunity for us if we can successfully develop these alternative delivery routes. Back over to you.
Thanks, Bob. Okay, so many of you are obviously asking yourselves, which of these approaches will work for my child's mutation? So this table shows which approach is relevant for which category of mutation. If you have your child's genetics report, look it up to see exactly what the mutation is. If you don't have the genetics report, but you know that it's been done, please ask your doctor for a copy. It's really important to have it. Now, point mutations are those that are caused by a typo in a single nucleotide base. For example, T158M, R306C, R133C. For point mutations, everything we discussed today is relevant, with the caveat that for RNA editing, it's only the G to A mutations that can be edited. Nonsense mutations are those that end in X, R255X, R270X, R168X. Those are examples. For these mutations, everything that we had discussed today is applicable, including the RNA edit. For insertions and deletions, all of those approaches are relevant, with the exception of base editing and RNA editing. And finally, C-terminal mutations. These are mutations at the end of the protein. And in these cases, the protein does its job, but the mutation at the end of it destabilizes it. And so it degrades and there isn't enough of it around. With funding from RSRT, Adrian Bird is working on a base editing approach specifically for C-terminal mutations. Now, we envision scenarios, as Bob mentioned earlier, where multiple genetic medicines can be given to one patient. For example, a gene therapy that's delivered virally, and then an RNA editing that doesn't need viral delivery could be given to the same patient. Having non-viral delivery options would really open up the door to giving patients multiple genetic medicines if needed. And we see this happening today in the SMA field. Please note that this graphic here is available on our website. Click on Cures, scroll down, and you should be able to see it. And if you have any questions about your child's mutation, you're not quite sure how to interpret it, you're not sure what applies, please reach out to me. My colleague's gonna put my email address in the chat. Feel free to reach out and I can help you figure that out. Now, this is a timeline that shows where we predict these various approaches will be by 2028. Hopefully there'll be a gene therapy that's approved by then, but we think that we will be in clinical trials or close to it for all of the other various approaches. Now, something that dawned on me while I was looking at this graphic a while ago, when RSRT launched in 2008, with the exception of gene therapy, none of these other approaches had been discovered or engineered, none of them. Think about that. I mean, I'm just amazed by that. And it just shows that we're really in a genetic medicine revolution. Bob, anything you want to add?
Yeah, like this gray hair came from something. I've been doing this a long time, since before the inception of gene therapy. And I'm so glad to be associated with Jana and Randy on the team to help successfully translate this work. And I recommend you spend a little bit of time on LinkedIn checking us out. And you'll see we understand how to move research into products. So I've never been more excited about those real opportunities. The progress and the pace of progress is really unprecedented. So in this field, people are moving from concept to practice in months, not years or decades. Lots of green lights ahead of us on Roadmap 2.0. It's time for pedal to the metal. Let's do it. Thanks.
We agree. Okay, so now I'm going to draw your attention to the cloud, our tracking initiative. And I'm going to turn things over to my colleague, Dr. Jana Van Heen.
Hi, everyone. Thanks, Monica. As Monica just mentioned, we have a very important effort in moving new curative therapeutics into the clinic, but we also want to support those programs once they're in the clinic and continue to support the existing ones. So as a drug developer myself, I'm going to tell you about the efforts we've been spearheading to support pharma to make it easier for them to choose Rett syndrome when they're trying to determine what disorder to invest in. So we're systematically checking items off the list that pharma companies need in order to feel comfortable starting a Rett program. So first, I'm going to talk about two very important sources of information that we're now using to help design trials: the Rett Syndrome Global Registry and our Citizen Medical Records Study. Then I'll talk about our biosensor development effort and our upcoming Vibrant study, which aims to measure symptoms better. And then I'll close with just a brief update on our biorepository, which is our collection of Rett patient cell lines that are accelerating research. A few years ago, when we were searching for what else can we possibly do to help cure Rett syndrome, we looked at the research landscape and we realized that there were two huge sources of information that could speed therapeutics that were just not being utilized: parent knowledge and medical records. Parents who care for someone with Rett syndrome have a lot of experience dealing with it. And if we could pool that knowledge, we would have the power to make it actionable to support drug development while enhancing what we know about Rett at the same time. So we decided to establish the Rett Syndrome Global Registry. This study collects information from parents about what it's like to live with and care for someone with Rett. Lots of questions about symptoms, how they change, what medications and therapies are used, where care is received, and many other questions that help not only pharma companies, but other parents as well. We worked really closely with pharma companies to make sure we were asking the right questions to help them design their studies better, understand what matters to parents, and maximize how to support their programs through the registry in other ways. We designed the database just like you would for a clinical trial. And we met with the FDA to make sure we could use the registry information collected to support drug development. I'm happy to say the registry launched last November, and the more patients we have represented there, the stronger the data set becomes. So please join if you haven't yet. Please update your information if you haven't been in a while, and use our Invite a Friend feature to invite other Rett parents. The registry is available in 10 languages, and it doesn't require anything other than an internet connection and some of your time. There are no clinic visits. And we currently have parents from 57 countries in the registry now, and we'd like to keep growing.
I'm just going to mention that we're going to put the link in the chat. So if you haven't joined yet, click on the link and please join.
The second study we're running to help companies is our medical records study with Citizen Health. This study turns medical records into something usable and comparable across all Rett patients who are participating. This is also a fully online study that takes advantage of all of the medical care your loved one is already receiving, and it doesn't require any clinic visits either. It's comprehensive in that it takes in all of the medical care at doctor's offices, hospitals, ER visits, so we're able to get a full picture of what the care journey has been, and it's limited really only by how far back your records go. Now, normally for natural history studies where you go to the clinic to participate, the study will collect information going forward, and a 10-year study will take 10 years to complete. But by looking backwards and making medical record data into something usable, we can build a study that would normally take decades in just a few months. And new medical records get added as normal medical care progresses. Some other advantages are your records are consolidated into one place. So if you need to reference them, if you have a new provider or you're in an ER somewhere, you can share this information with those doctors. And the data captured by Citizen for another rare disorder, not Rett, has already been used to support a drug development program. So there's a precedent that Citizen natural history study data are acceptable to regulatory authorities, which is not normally the case. And if a company were creating these data sets, they would be proprietary and couldn't be shared. But since these efforts are going through us, they have the chance to help everyone. And that's our goal. And one last critical point, Citizen Health licenses anonymized data to companies, which helps them design their clinical trials better. But they recognize that this resource wouldn't exist without patients. So part of the licensing fees get returned to participants. That's right. So if your loved one's data gets licensed, you will receive a payment. And some parents have already received $1,000 just for signing up and letting their medical record data be used for research. So based on data privacy and sharing laws, Citizen can collect medical records on your behalf only if you're in the US for right now. So if you're in the US, please join. If you're outside the US, you would just need to upload your own records. So either way, please consider joining.
And the link will be in the chat for how to go about joining. It's very simple, only takes a couple of minutes.
Okay, let's talk about our biosensor development initiative. This is a really fun program that has huge implications for how therapeutics will be tested in the future. So let's just start with traditional outcome measures. So in clinical trials, you collect information on the symptom before treatment, after treatment, and then you compare them to see what happened. Now in Rett syndrome, like other brain disorders, we measure symptoms through rating scales, or sometimes they're called questionnaires. And this is where someone, a physician or a parent, will answer the same questions about the symptoms of interest at different times during the study. In Rett, we rely on parents and doctors to rate symptoms because Rett patients can't rate themselves. So we're limited right now to making observations rather than measuring anything directly in the patient. Here's some examples of the most commonly used rating scales in Rett. The top panel is the CGI-I, or the Clinical Global Impression Scale of Improvement. And this is used by doctors to rate Rett syndrome overall in an individual. They use a scale of 1 to 7, covering various change from no change to very much improved or very much worse. In the second example, this is a rating scale that parents would complete. 2 questions are shown here for sleep and breathing out of the 45 total questions on the scale. This is the Rett Syndrome Behavior Questionnaire, or the RSBQ. And in this case, parents read the statement and then they rate their response as a 0, 1, or 2 based on if the statement is not true, sometimes true, or mostly true. Now, what you probably thought when looking at these scales is that the answer is going to depend, right? It's going to depend on who's answering the questions, what they've seen, what they were told, what happened yesterday versus 3 weeks ago. And so these scales, simply by human nature, are influenced by biases we as the observer have, even if the patient's only getting placebo. Often these rating scales come from different disorders, or they're not used for the purpose they were created. And in the many studies I've run over the years, in most cases, one requirement was that the rater of the scales can't change during the study, because different people will rate the same patient differently, different doctors, even different parents of the same child. So the goal of our biosensor development program is to make it easier for companies to include biosensors in their trials, to generate cleaner data on fewer patients by measuring symptoms directly in the patient and consistently. Let me show you what I mean. Here's an example of some biosensor data we've collected in the last few studies we've run. Now, without any background, if you just look at these 2 panels of data I'm showing here, even if you know nothing about what you're looking at, you can see clearly that these patterns are very different between the 2 groups that are shown, and that's the point. What you're looking at here is actually biosensor data captured over 1 night. Breathing is shown as a green line of inhalations and exhalations in the left, and you can clearly see that the healthy breathing at the top is very consistent and even, and on the bottom, you can see that Rett breathing is not that. Changes in heart rate overnight are on the right, and we can see that Rett patients that are more severe, based on a clinical global impression, shown in red, are not the same as the patient group that was rated less severe, shown in blue. If we look at sleep staging over 1 night in a healthy individual and a Rett individual, we see there are vast differences there too. And what these types of data allow us to do is to begin to precisely measure symptoms. We can move away from a handful of answer options or wondering what our answer depends on. We can identify different sleep stages and breath holds and other things by analyzing the signals in the same way every time. And we can count them and we can measure how long they last. And that kind of information adds way more depth to understanding a symptom like this than a rating scale. And if these symptoms, which don't require any learning or skill implementation, correct faster in a trial than, say, hand function or communication, then we now have an early predictor of later improvements. And that's very valuable. So if we can get biosensors into clinical development, we need fewer patients over shorter periods of time, which means faster approvals and faster access to these new medicines. Okay, so I'm about to just start our latest biosensor study called Vibrant, which stands for Validating Innovative Biosensors for Rett Autonomic Symptom Tracking. This study will be conducted at 3 centers in the US, Boston Children's, Rush University, and Texas Children's Hospital. And the point of the study is to show that the biosensor devices we're testing are reliable and accurate compared to the tools a doctor would use in the clinic. And for this study, that's specifically an in-lab overnight sleep study, which is considered the gold standard. We're collecting heart rate, breathing, blood oxygen saturation, and movement from Rett patients at various times over 9 weeks. And the study will build confidence in these devices, which makes them more attractive for use in clinical trials. And some of our work in this space has already led to biosensors being used in trials. So we want to keep that momentum going. Our effort to make biosensors an option for companies is really important because it can accelerate all of the different clinical development programs. And most of the companies working on Rett don't have the resources or the bandwidth to do this themselves. Here's just a quick look at some of the devices we've been working with. You can see the O2 ring on the left for oxygen saturation, the Hexoskin shirt, the RespiraSense breathing patches in the middle that measures breathing rate, and then the invisible or the non-wearable device called Emerald, which actually doesn't touch you at all and can assess movement, sleep, and breathing from a distance, is shown on the right. Okay, and to close my section here on our biorepository, this isn't really a clinical tracking tool, but it is a very important resource to help companies explore their therapeutic candidates in human cells from Rett patients. I want to take a quick moment and just thank the families who participated in a study with us and donated samples to help create these collections. These samples continue to be an important, valuable, and sought-after resource that is really moving research forward, so thank you so much. We have 2 collections of patient cells, a skin cell collection at Sampled and a stem cell collection at Coriell. The stem cell lines, called iPSCs, can be used to make brain cells in a dish, and this allows the testing of therapeutics in the exact human cells that we need them to function in. And this is another resource that companies don't normally have access to. Here's a breakdown of who's ordering the lines and what they're doing with them. About a quarter of our requests come from companies. You can see more than half of the projects are focused on cures, about a quarter on understanding more about MECP2, and 11% looking for drugs that might improve characteristics of Rett. So thanks for listening. With that, I'll turn it back over to Monica.
Thank you, Jana. Okay, so I mentioned in the beginning that Roadmap has a $40 million budget, and Tim is going to provide a little bit of context around where the funding comes from to RSRT.
Hi, everyone. I'm Tim Freeman, Chief Development Officer at RSRT, and I am the proud father of a 15-year-old daughter with Rett syndrome. And my role at RSRT is to work with you, parents, grandparents, and families to fundraise for this research. And fundraising by Rett families is critical to making this bold research plan possible. It simply cannot happen without your action. And I'm not talking just to US families here too. Rett syndrome is a global disorder. We need families from all around the world to get involved. I'm going to start with 4 important key points. 1, fundraising isn't scary. Anyone can do it. And it can even be fun and fulfilling, believe it or not. 2, money makes this research happen. That is the simple fact. If all of us do this, it grows funding exponentially. It speeds the research towards the cure that we all want. 3, yes, my role at RSRT is fundraising, but the power to raise this money does not lie with me. It lies with you. And I'm here to encourage you and help you to raise funds. And 4, if you remember nothing else from the words I'm going to say here, I hope you'll act on one thing. And that one thing is to contact me by email or phone. Maybe you want to jump in and start making a difference right away, and I hope you do. Maybe you have ideas about fundraising and you want to brainstorm. Maybe you disagree with me on something that I'm going to say here, and that would be, I'd love to hear from you. I'm here anytime to talk with you about all of that. So I want to give you a sense of the scope of our recent fundraising at RSRT. Over the last 5 years, we've averaged about $8 million a year in donations. it is definitely going to be a stretch for us to raise 10 million a year over the next 4 years to carry out this research plan that you've just heard about. $10 million a year is a 25% increase from our usual fundraising. So it's going to take a big effort from Rett families to make that happen. I'll tell you more about that soon. So where does RSRT's funding come from? I think it's important for everyone to understand this. This chart shows the percentage of our donors that can be traced back to a Rett family. So 99.8% of our supporters, virtually every donor to RSRT is connected to a Rett family. And I want to be very clear here. This does not mean that all our donations come from families that have a child with Rett syndrome. What it means is that every donor knows a Rett family or is related to one. And it shows that the only thing that results in donations coming in is when Rett families take action and ask their networks to make donations. And I'll add too that this is not unique to Rett syndrome or to RSRT. Funds raised for research on any diseases, especially rare diseases, only come in when a family that's affected by that disease takes action and asks their network to support. So what are the sources of our funding at RSRT? 90% of our support comes from donations made in the US, 10% comes from international donations. And how are these funds raised? What actions do families take that lead to funds coming in? So about 60% of our funds come from events, all of them spearheaded by Rett families. 30% comes from donations and pledges that are unrelated to events, almost all from Rett families. And about 10% is from crowdfunding campaigns that are done on our platform or on Facebook or other social media. All of those campaigns launched either by a Rett family or someone very close to a Rett family. So the further makeup of that funding is 89% of our support comes from individuals, people, 7% comes from private foundations, 4% from corporations, and we get no funding from the government or the NIH. And I do find that people are often a bit surprised by what a small piece of the pie corporations are, but that is not unique to us either. Companies make up less than 10% of all philanthropic giving to all causes in the US. So the point here is that individual donors make up the vast majority of funding across the board to all causes and to RSRT as well. So how are we going to raise this $40 million in 4 years? We need you. We need Rett families to take action. So now we're going to get into the heart of this. What actions can you take that will result in your network supporting this research? And there are really two major ways that you can fundraise. One is crowdfunding, and the other is starting an event. And let's start with crowdfunding. So we have our own crowdfunding platform at RSRT. It's called RettGive. It's at rettgive.org. And there are several big advantages to using RettGive. First of all, it's super easy. All it takes is emailing me some materials, and I can set up your campaign for you. Second, it costs nothing. It costs nothing to you. It costs nothing to RSRT. Third, the funds you raise come directly to RSRT, directly to the research. And because donations come right to RSRT, it gives your supporters confidence that they know where their support is going. You can also start a campaign from anywhere in the world, and it can be in any language. So again, this is especially terrific for international families. And finally, the campaigns are personalized. They have photos of your child and your own message that you've written. And on the right-hand side of your screen here, you can actually see an example of a RettGive campaign. This one was launched just about 5 days ago by a dad who's in Atlanta, Georgia, and he's doing it around a road race that he's running. And you can see that he's already surpassed his goal of raising $5,000. So it's just a demonstration of how quick and effective and easy this is. That's $5,000 that this dad raised in just a few days. And that makes a difference to this research. And some families do campaigns around runs like he did, or climbing a mountain, or a birthday, or an anniversary. But there are plenty of families who just start a campaign. That works just as well. It doesn't have to be tied into an event or an activity. So how do you start one of these campaigns? Again, I'll set it up for you. To everyone who's on this webinar, I will send all of you the instructions for the materials to email me, which include some photos and a short paragraph that you write. And I really hope a lot of you will give this a try. I think you'll be really pleasantly surprised at how easy it is, how fulfilling it can be, and how enthusiastically your friends and your family will contribute. So another way to raise funds for the research is to start an event. And I want to dispel a few myths that are out there about events. First of all, an event does not have to be a big gala. We have a huge range of events and what they look like. We have backyard barbecues and walks and happy hours in bars and restaurants, cocktails in your own home. And yes, there are also big galas. The point is, an event can take any form you want it to take. Another myth out there is that you need to have a network of wealthy friends in order to hold an event. And that's not true at all. People at all income levels and all demographics come to events and make donations to them. And families at all income levels can start an event. And the last myth that I want to dispel is that you have to have experience with events or fundraising to start an event. And not at all. Anyone can do this. Reach out to me. I can work with you on it every step of the way, and it's be easy, and it's enjoyable and effective. If you live in an area where we already have an event, an easy way to jump in is to join the committee of one of these events. These are our upcoming events that you see here. So being on a committee gives you leverage to reach out to your network. If you live anywhere near any of these events that are coming up in the fall, please contact me. We can talk more about your joining the committee. These events are fun, beautiful, poignant. It's a chance to meet other families. So I really do encourage you to get involved. You know, I'll add one other way that we've raised funds at RSRT is by working with families on a tailored approach to someone they know, to a contact of theirs. So maybe you have a connection to someone who has a foundation or is especially philanthropic. So please contact me if that's the case. I can work with you on a personalized approach. It would be an approach that is comfortable for you. Maybe it's a letter that I help you craft from you. Maybe it's a letter from Monica or from me. We can come up with a plan together that works. So fundraising for this research is it's really one of the most important things you can do, that you will ever do. Not only does it advance the research for all our loved ones, but it puts you in the driver's seat. It means you are taking action for change. And I have found that can feel very, very empowering. And this research plan that you've heard about today, it just cannot happen without all of you being bold yourselves, getting out there and getting involved and fundraising. So I hope very much that you'll contact me to get started. So thank you very much.
Thank you, Tim. Okay, so we told you today about our plan, Roadmap to Cures. The goals are to select 3 genetic medicine programs, advance them to clinical trials by 2028. We discussed that developing genetic medicines is an iterative process, and that the first genetic medicine out of the gate is not going to be the last or the best. It'll be the first. And now you know that because Rett is not low-hanging fruit for companies, they are unlikely to start a genetic medicine program for Rett from scratch. And finally, I shared that together we need to feed the pipeline, and that cures for Rett are dependent on keeping this pipeline fully stocked. Now, the RSRT team knows how to do that. We're putting together all of the stakeholders to get us to cures: the academics, the biopharma companies, the regulators, the consultants, the RSRT team, and you, Rett families. You've heard today what a critical role you play. I hope that my colleagues and I have convinced you that RSRT is worthy of your support and in great need of your action. So let's talk for a minute about a small word, just 4 letters, but a word that is packed with so much emotion, so much hope, so many dreams: the word cure. And I get a little emotional myself because this is so personal to me. Now, there are disease groups, companies, scientists who shy away from using the word cure. They don't want to provide false hope to families and to patients. They're worried about overpromising and under-delivering. At RSRT, we are unapologetically going after cures for Rett. That is our goal, and we're not going to stop until we get there. Now, I'm going to just stop sharing my screen for a minute so you guys can see me a little bit more up close and personal. What I am not promising, and I really want to be clear about this, is a cure for your child. And that's because we don't yet know what symptoms can be reversed, by how much, at what age, what mutations, what severity. Can kids with full-blown symptoms be cured? Like it never even happened kind of cure? Or will the genetic medicines need to be given at birth or before birth in utero? We're going to start getting clues from the current gene therapy clinical trials. What I know is that all of the data I've seen points to the fact that our children can improve. I've not seen any data that diminishes my optimism. In fact, I recently said to a colleague, if parents knew what I knew, they'd be filled with so much hope. Let me go back to sharing. Now, my daughter's 27. I know that there won't be a full cure for her, but I do believe that her symptoms can be dramatically improved. I think all of us would take dramatic improvements over the current status quo any day of the week. Now, about 10 years ago, I went to a scientific meeting organized by Huda Zogby, full of academics and biopharma scientists. At the end of the meeting, I posed a question. If Bill Gates had a child with Rett, would we have a cure by now? The consensus was no, we would not. Gene therapy to the brain was still science fiction. The other technologies didn't exist yet. There was just still so much science that had to be discovered. And that would take time, not just money, and no amount of funding would change that. Today, we are in a completely different situation. We have the technologies. The scientists are in place. We have biopharma's attention. The main obstacle is money. Money will let us move more aggressively and pursue all of the approaches that you heard about today and the future approaches that you can bet your bottom dollar are coming. Money will get us into more companies. Money will make things go faster. Now, there are hundreds of thousands of families around the world with a child with Rett. If a fraction of us, say 5,000, raised $10,000 a year, we would have $50 million a year for research. Can you imagine what we could accomplish with $50 million a year? So please, let's not wait for some billionaire with no skin in the game to come and save our children, because it's not happening. But the good news is we can all step up and we can save our children ourselves. Okay, so now it's time for Q&A. So I was really concentrating and I haven't looked at any of the questions yet. So I'm taking a look at your questions for the first time now. So I'm just going to start and work my way through them. We have 15 minutes. If we don't answer your question, I promise that we will email you. And if you don't hear from me, contact me with your question. We want to answer all of your questions. So Jordan says, will gene therapy be helpful for those with atypical mild forms, such as those with mutation R133C? Do the benefits outweigh the risk for someone with a mild type? So I don't see any reason why individuals with mild mutations shouldn't be helped by gene therapy. But right now I'm answering that in a vacuum because I really haven't seen results from the gene therapy trial. So let's wait to see what the first results say, but there's nothing for now that I know that would make me say that mild patients wouldn't be helped. My colleagues, please jump in if you have anything you want to add to that. Okay. Let's see. If a child is in a clinical trial and receives an early treatment that hopefully helps, but there is still room for improvement, can the child receive additional treatments that would develop later? So we addressed that in the presentation. It depends on what the delivery technology is. For now, if there's multiple genetic medicines that are all being delivered with viral delivery, you would only be able to get one one medicine delivered virally. That will change if the genetic medicines are delivered with non-viral, or if scientists figure out a way to do something to the viral vector that makes it stealth and evades the immune system. And again, please jump in if my colleagues want to.
No, it's correct, Monica. Using AAV has the risk of inducing a severe immunological reaction, so that a second dose would result in anaphylaxis and severe risk. So it's about the body's immune system rejecting AAV. That's the complication for the one only opportunity with AAV.
Do you have so Nora says, do you have an estimate of cost for the upcoming treatments? Well, we know from other disorders that have approved genetic medicines, these are not cheap medications. These are multi-million-dollar gene therapy, but you've also heard from Bob that the cost of developing a single dose is $1.5 million. So no wonder these genetic medicines are expensive. Now, companies would not have a business model if they expected parents or family, patients to pay for these out of pocket. So, as they're moving towards commercialization, if commercialization is warranted, because of course it all depends on the results. But as they're moving towards that, in parallel, they'll be having conversations with payers, with insurance companies, with government, Medicare, Medicaid. They'll also be pursuing approvals outside of the US and doing whatever it takes to get that done. So they'll be talking to the regulatory agencies in other parts of the world. And the goal for them will be to get payers to pay, not families. There's not going to be enough families that can afford this, to provide a business model. Okay, let's see. Can IT, okay, Jennifer asks, can IT delivery be delivered to people with scoliosis? So the answer is yes. It is publicly known that one of the patients had scoliosis that received IT, but it's a case-per-case basis. So I guess it depends how the surgery was done, where it's done. So it's case by case, but the general answer is yes. Let's see. So Michelle says, my daughter was diagnosed at 2 years old and now at 2.5. Honest thoughts. Will she eventually benefit from gene therapy and or treatment soon in her young lifetime? Thoughts on how far away, as long as something goes well. I think she means maybe outcomes go well. Well, you know, the gene therapy trials have started. Let's see what their results are. They're on track. If things are good, they should be approved, you know, around 2028. Our programs that we're moving forward will be in clinical trials by 2028, and hopefully things will go smooth and things, you know, those programs can be commercialized as well. So I think your daughter's going to have options. And I know it never happened soon enough. You know, I've been doing this for 25 years. I never thought it was going to take 25 years. So I feel your pain. Please know that we're working as fast as we can and that things are moving more rapidly now. I mean, the good thing about science is it's not linear, right? There are things that can really move things forward more quickly. So my daughter has 2 mutations. Oh, that's interesting. R168X and R294X on the same gene. Does this cause any obstacles for these treatments being discussed? I don't know what that means, Angela, to try to get your daughter into a trial. I don't know whether having 2 mutations. I mean, it's Rett syndrome. It's not mutations in another gene. So I don't know. But in terms of receiving these medicines once they're approved, I don't see any reason why your daughter wouldn't benefit from it.
Monica, just to add to that, if there was a mutation-specific therapeutic, you would have to target each of those mutations separately, but something like a gene therapy that replaces the whole gene would cover that. So there are still options for your child.
The generic ones are gene therapy, eventually prime editing, RNA trans-splicing, MECP2 reactivation. What's going to be so Becky asks, what's going to be the difference for our younger versus older kids and adults? I think we have to wait and see the results. I think, look, common sense suggests that the earlier you can intervene, the better. Is it better to intervene in a 1-year-old or a 30-year-old? I think we all know the answer to that. But Rett is quite unique if we look at the animal data and the reversibility studies. Very old mice who have never had MECP2 and have severe Rett, they can have their symptoms reversed just as dramatically as younger mice. So now people are not mice. And so we're going to have to wait to see how that translates. But the science bodes well. And I, you know, like I said, I don't expect a cure for my daughter, but I do think dramatic improvements is not unrealistic. Feed those pipelines. Yes. If a different viral vector is used, could a girl then receive 2 viral delivered treatments? Probably not. Viral vectors, even though they're different, they're kind of in the same family. So unlikely. We would need viral and non-viral or all non-viral. Or small RNA, like the RNA editing is kind of its own category. Why focus so greatly on viral deliveries if they seem cost prohibitive? Yeah, that's a really good question, Catherine. It's because that's what we have right now, right? If we wait for non-viral, we don't have non-viral delivery yet that's proven to get into the brain. So we could put everything on hold and just wait, but we don't want to do that, right? Because our kids need something now. And so we're moving ahead with viral delivery and we're doing everything that we can to, you know, understand the non-viral landscape, to invest. We just made our first $1.5 million investment just a couple of weeks ago. We'll be doing more of that. And then when there is a breakthrough, us and everybody else that's working on neurological diseases is going to switch. But I don't think putting things on hold and not doing anything while we wait is the right approach. Let's see. Yeah, I think I answered the mild versus severe. Can someone who lives internationally, who's over 18, join the clinical trials in America that are currently recruiting? You know, all the clinical trial sites are public information. You can Google clinicaltrials.gov and find them. You're more than welcome to reach out to them. My feeling is that they're going to recruit locally from patients that they know. And one big reason for that is that they're going to try to not disrupt a patient's life, right? So if a patient is local, because you need to stay close to the clinic for a number of months so that they can monitor you. And if you're local, that means you have your therapies, you're living in your home, you're going to school, you're doing all the things you normally do when it's less disruptive. And with any clinical trials, you're trying to control variables. And so my feeling is that's what they're going to do, but you can certainly contact them. So David asks if R133C is a point or C-terminal mutation. It's a point mutation, David. What are we doing in time? Okay, so Elizabeth says, you mentioned that RNA editing can be given with a gene therapy. Does this have to be simultaneously, or can a patient receive gene therapy and then RNA editing treatment years later? Now, it would not be simultaneously, Beth. It would be, you could get first of all, they're not going to be commercialized and available or probably in clinical trials at the same time. So you could get one and then you could get the other one. David asks, can current financing environment for biotechs stalled exciting opportunities? Any green shoots, or will it still be a few years before uptick? I'm not sure. Okay, so I think he's saying, so it's a tough climate right now for companies, for biotech companies in terms of investments. It's very difficult for them to access capital. And I think David is asking whether that's affecting us. Well, Bob, I'll be interested to hear I think it's actually helping because they're likely more interested in speaking with us about potentially funding Rett programs, because it's a more difficult time for them to access capital in other areas. Go ahead, Bob.
Yeah, I think so. I think with the recession over the last 2 to 3 years, there certainly was a stall or a delay in new companies being able to advance programs like Rett. I think we've been out of the woods for about a year, and I agree with Monica. We're getting some very attractive proposals from people who would like to use Rett as a proof of principle, and working with us see a path to commercialization. So it's not a yellow light, it's turning green.
So Isabel says, what's the plan to ensure therapeutic development benefits people outside the US, especially in lower income countries? Yeah, that's a really good question. I mean, companies, so one thing that I think we made clear, but worth saying again, our plan is not to take a genetic medicine program all the way to the end and commercialize it. And, that takes a lot of specialized skills and people and money. And so our hope is to generate the data, de-risk these programs, find a company to advance them, similar to what happened with Taysha and Neurogene, right? We want to do that over and over again. And then of course it's up to the company to commercialize and decide where they're going to go. I think all of these companies are going to go to Europe and other parts of the world. Now, low-income countries where there isn't going to be a payer who's going to pay out $1 million, $2 million, $3 million is going to be more difficult. That's why we're really hoping that non-viral delivery becomes an opportunity, because then I think the cost dramatically comes way down. And also because of the ease of making it, it's going to be just easier to get access. Does anybody do my colleagues want to add anything?
The only thing I would add is that it's the company's decision. We don't really have control over that, but we can try to raise awareness and raise the need to try to get them to focus on pathways and opportunities for people in countries of low income.
Okay, here's a lot of questions, but they're good questions from Millie. Great presentation, thank you. From my limited knowledge, a good example of distributing funds to all kind of treatment options is the cystic fibrosis community. Are we modeling any of our strategies after their strategy? Yes, we are. I know Bob Bell, who was the CEO of the Cystic Fibrosis Foundation for many years. He retired a while back. one of the first people I reached out was when my daughter was diagnosed, was the founder of the Cystic Fibrosis Foundation, Doris Tulson, who lives in White Plains, New York. She's well into her 80s, maybe she wouldn't like me saying that, so I won't say that. But she also retired recently, but she was a real go-getter. And yes, we've been following the strategies of the Cystic Fibrosis Foundation. They have $3 billion in their bank account. So when I say that money makes a difference, this is kind of exactly what I'm talking about. When the Cystic Fibrosis Foundation makes an investment in a company, funds a program for cystic fibrosis in a company, they're not giving $1 million or $2 million, they're giving 15 million, 20 million. And that's a big mind share from that company. That's a program that they're really going to prioritize. We can't do that. If we could, I think it would make things go faster. So that's really an example of what I'm saying. Your other question is, why only 3 programs? Why not disperse the 40 million to numerous different programs? Well, we used to do that when we first started, small grants, $50,000, $100,000 grants to lots of people. That doesn't get you very far. I think that's okay for basic science where you're seeding the field, you're trying to get lots of people to work on Rett, and we did that years ago. But I think now we're at the stage where we need to, we're doing translational work, we're doing genetic medicine drug development, we're not funding basic science, and that takes money. You give smaller amounts of money, it's not going to get you very far. How confident are you that 40 million will get us to 3 different clinical trials? We're very confident. What are the pitfalls? Bob, I'm going to let you answer that.
Probably just the time that it takes to go through safety and toxicity studies. At this point, we understand that these different modalities can work, and it's about delivery and then addressing any unanticipated safety issues. And we're very, very confident. Our pharma partners, ProQR as an example, is putting money down that they're going to be in the clinic in 2027. And we've looked at their development plan, we critique it and build with them, very confident that they're achievable goals. We have several other partnerships that we're dialoguing that aren't public yet, that would help you understand the confidence we have in being able to succeed here. And as I said before, we know how to do this. The team here understands how to take concept from academics into the clinic.
The final question, she says, is with Citizen Health, do you need to manually upload all the medical records, or does it automatically sync with something like MyChart? You don't have to upload anything if you live in the US. You just open an account, provide informed consent, few other little steps. Literally, it takes you less than 5 minutes. And then Citizen Health goes out and gets all the records because you've put in your child's doctors, clinics, hospitals, wherever they go. They do all the heavy lift. You don't have to do anything. So please do it. I've already gotten 2 checks totaling $1,000. That's nice too. I bought Chelsea some nice things with it. So please do it. And if you're in the UK, or not in the UK, I just read a question that said UK. If you're outside of the US, then yes, you have to upload your medical records for now. How are we doing on time? So actually we're done. I'll take one more. Go ahead, Bob. Did you want to say something?
I wanted to give people on the webinar a chance to thank you and for everything you've done, giving such a wonderful talk and motivating and concerting such a talented group of people. You're just really a star.
Thank you.
So watch the hearts. Come on, those hearts need to go up. Let's go.
Hearts. Thank you, everybody. Well, listen, this has been fun. Our plan is to do this more often. We get so caught up and so busy and just working that sometimes it's hard to carve out the time to plan a talk and to do it. But we wouldn't be here without you guys and your efforts and your fundraising efforts. And we will do a better job at keeping you updated. So thank you, everybody. I'll get, if we didn't address your question, we'll do it via email. Have a great rest of the week.